This is a software package developed specifically for phylogenetic analysis, so 16S sequencing type data. These are all the tools around that kind of thing. And of course, QIIME is one of the most commonly known pipelines for analyzing 16S data. It kind of brings these things together. So as we were analyzing our own data, we kept running into roadblocks or just things that we wish were different. And so we kept writing our own code to work around these things or just add things. And we eventually just organized them into a single package to provide parallel but yet intercalating tools for analyzing 16S data, mainly with the QIIME pipeline in mind, but where possible we use open interoperable data sets, from simple CSV to, and now I'm blanking on the term, but other open standards for data where possible. So this was, what I showed you here was the basic pipeline for QIIME. These are the steps that we added or modified to that main pipeline, and I'll just quickly run through some of these. We had published previously on primer averaging that if you sequence with multiple primers, you get better results because certain primers, at least in certain environments, certain primers have biases towards certain organisms versus other organisms. That is something that if you're willing to sequence twice, which nowadays isn't so much of a big deal since sequencing has dropped in price so much, you can improve accuracy of your results. The other problem that we noticed with QIIME, especially with species-specific analysis, which we do a lot in the oral microbiome, is that QIIME tends to give you these OTU results with 50 different versions of strep mitis or something, which can be useful if you're interested in different subtypes of bacteria. But for certain things like visualization, certain visualizations, it's better if we only consider one of those per entry. So we provide some facilities to condense those down into a single representative OTU per species. We also have a bunch of programs for either slightly better or alternate visualizations of a lot of the data. One of the most commonly known for 16S data is the principal coordinates analysis, beta diversity kind of thing. So this is the default QIIME plot. They have since released their Emperor tool that provides a 3D in-browser visualization. But if you want 2D visualizations, you still have to go back to the old way and do this, or do sort of a flat two-axis view of the 3D data. So we have a tool that provides both within the same script based on, and sorry, I forgot to mention at the beginning, this is all Python. So this is all based on matplotlib in Python. So you can specify either 2D or 3D within the same tool, get the same results, and it'll automatically, for example, in your mapping file, it'll take it'll automatically figure out all the categories in a column that you specify. It'll take colors. You can specify a column of colors in your mapping file, and it'll do that. We provide a bubble plot version of this. So you get one plot for each species or genus or whatever you want to specify, and it sizes the plot points in the PCOA plot by species relative abundance, which here we used, we published last year, we used to look at specific species, pathogens versus non-pathogens in various environments, and we could see some significant differences. The other nice tool that we added is automatic production of data necessary to put into ITOL, which is a very useful tool, but requires you to make all the data sets yourself, and QIIME provides no utility to do that. So given just the end product, the species or the OTU abundance table produced by QIIME, that goes input into our program, and we can produce all the data sets necessary to input then into ITOL to produce nice looking differential analysis of species in phylogenetic trees. These are just some examples of different projects that we've done. And this is the last thing I want to touch on is something very recent. So this is an alternative dimensionality reduction technique that isn't used often in species analysis, but we've noticed it tends to do a slightly better job than the PCOA, so linear discriminant analysis, which we have a script now implementing that, again in Python. And then again, this is very new, but we can take the linear discriminant analysis, throw it into k-nearest neighbors, and get nice partitioning of your groups. That's what you want. So the source is open up on GitHub. We have documentation hosted by Read the Docs, which is excellent. And it's available free and open source with an MIT license, all Python. So those are the things we've added, things we've done. My mentor, Dr. Kumar, various other people who are involved in collection of data and suggestions and our funding. So, thank you.